Our next speaker is Doug Richman, who is a distinguished professor of pathology and medicine here at UCSD and the Florence Seeley Reiford Chair in AIDS Research. He's the director of both the UCSD Center for AIDS Research and the AIDS Research Institute. Doug's laboratory was the first to identify HIV drug resistance and joined two others in identifying latently infected CD4 cells as an obstacle to the eradication of HIV with antiretroviral therapy. His lab is currently developing assays to measure the latent HIV reservoir in various compartments of the body and in various subsets of CD4 lymphocytes in order to characterize the targets of intervention strategies and to measure the impact of these interventions reliably. He's also the PI of the neurovirology core. So thank you, Doug.
Good morning. I'd like to start off by congratulating Igor and Bob and all the members of the HNRC and HNRP. Wish them a very happy birthday. It's been a remarkable 25 years of watching sort of the incipient ad hoc collaborations 25 years ago turn into a program like this, and it's been a very impressive accomplishment, so congratulations. So I was asked to sort of give an introductory overview of the cure agenda, which obviously involves, if one hopes to cure a person, we have to do it both above and below the neck. And so that's where we're, the focus is with this group, at least the above the neck part. And so HIV chemotherapy has been one of the really great medical accomplishments over the past 2 decades, dramatically reduced morbidity and mortality of those with access to care. I guess the number one really achievement that needs to be accomplished is the practical one of providing care to more and more people who need it. But in terms of the scientific challenges, I see really 2 major challenges that the field needs to address. For those who are uninfected, we need to develop a vaccine, which is the really most effective way to prevent transmission. And for those who are infected, it would be great to develop approaches to cure the latent reservoir. Why would we want to do that when we have such great drugs? Relieving treatment fatigue. I have patients who are now in their 3rd decade of suppression, who started on the original Merck 035 study, and they're hoping to continue for another several decades, but it would be great if they could stop. Reducing drug costs, reducing toxicity, their morbidity and mortality associated with viral persistence and immune activation, which we'll hear about later from Steve Deeks and others, with the associated complications, including the CNS impairment that Igor told you about. Reducing drug resistance and reducing transmission. So there's a lot of reasons why this goal would be great to achieve. But there are a number of questions which those of us in the field writing grants call research opportunities to address the HIV latent reservoir and eradication strategies. And we really don't know the answers to any of these. How is latency established and maintained? What are the cellular targets that are candidates for drug discovery to activate latent virus safely? This 3rd question is a little less controversial than it was several years ago, but do our current regimens completely suppress replication? What's the composition of the latent reservoir in terms of size, cell types, and so on? 5, are there anatomic sanctuaries, CNS, genital tract, and I won't say any more about the CNS since both Serena and Ron will be talking about that shortly. Will drugs that induce HIV transcription, the so-called kick drugs, result in I've got a typo in there already. Huh? In cytolytic death, or will an additional factor be required? There's a question about whether induction of transcription necessarily leads to induction of translation, so that the viral proteins are expressed so that the cell will either lyse or be susceptible to cell-mediated immunity. And then the question is, do we have to do something to enhance that T-cell immunity to kill such a cell? Will gene therapy or immunologic interventions contribute to this approach? And how do you measure the reservoir and the impact of candidate eradication strategies? So there's a lot of things that the next generation of investigators are going to have to figure out that us older folks have not succeeded in doing. In terms of different approaches to strategies for a cure, there's the quote-unquote kick and kill, which is eliminating a residual replication with antiretroviral therapy, and then eliminating the infected cells by selectively killing them off, either by inducing cytolytic replication or exposing the antigens for T-cell killing. And that sounds like a nice approach. But as with everything I'm going to say, it's all in a very rudimentary stage. And I think any chance of a true success, either for a vaccine or for this cure approach, will take a minimum of a decade. Enhancing HIV-specific immunity are things that people are looking at. The concept of a therapeutic vaccine, a concept that has no precedent, but is a nice idea, or enhancing the immune function. The ones that are currently in studies are inhibiting PD-1 or PD-1 ligand, approaches that have shown some preliminary promise in melanoma and other oncologic approaches. And then the final approach is even more Star Wars, and that is making cells resistant to HIV by gene therapy approaches, either targeting the provirus with zinc fingers or CRISPRs or so on, targeting CCR5, and there are trials that are in progress from a couple of groups, or targeting infected cells with some sort of toxin conjugate with a specific antibody conjugated to a toxin. So this is a diagram of the approach. You protect uninfected cells with antiretroviral therapy, and then you do something to selectively kill off, induce transcription, translation, and selectively kill off those cells that are latently infected. So the first sort of approaches that people have taken are with histone deacetylase inhibitors, which decondense chromatin and help induce transcription. Things turn out to be a lot more complicated. I won't go into the details. But the earliest studies from Dave Margolis and Sharon Lewin suggest very modest, less than a log induction of transcription, and really no measurable impact on the reservoir. The more recent studies with panabinostat are still show something happening, but once again, they're too modest to provide promise for something really beneficial. So we're going to need more than just one or two new drugs. We're in a very early stage, and there are several pharmaceutical companies, that, especially Gilead and Merck, that are have pretty intensive discovery programs to identify better and additional compounds that may be useful. These are data from the Merck group, in which they set up a high-throughput screened almost 3 million compounds with and without SAHA, which is a HDAC inhibitor combination. And they found 2,400 unique hits or compounds that had some activity in inducing transcription. And what's interesting, 16% were histone deacetylase inhibitors. They found a whole new class, farnesyltransferase inhibitors of 17%, which may or may not have any utility. And then 2/3 of the compounds they found are so-called orphan or unknown target compounds, and this is really the focus of identification at Merck and in the CARE collaboratories, trying to find out what they're targeting and how useful they might be. Some of the challenges to define and characterize the latent reservoir are going to require assays that are better than the rudimentary assays we've had. And these are going to be necessary both to define the reservoir, where is it in terms of what cell types and what anatomic sites, and we need assays to measure it so we can characterize the composition and actually reliably measure changes after candidate interventions. So that's some of the work that's been going on here with my colleagues at UCSD. And it turns out most of us do all of our measurements in the blood because that's the most readily available. But the blood, in terms of the latent reservoir, represents somewhere probably between 1 and 2% of the actual reservoir we want to measure. The rest exists in the body and in tissues, which is both less accessible and is not as well mixed and homogeneous as the blood. And Joe Wong, who's sitting in the audience, has done some of the nicest work on looking at the gut and documenting both some of the reservoir there and the fact that it's quite heterogeneous, and even where in the gut you sample will show different sizes and types of reservoir. So, as I mentioned, the measurable analytes in the blood represent only the tip of the iceberg. The extracirculatory reservoir is not homogeneous and difficult to access. And then finally, most published reports have inadequate validation of performance characteristics, something that is more typical of academic labs and not acceptable for registrational studies by the FDA and pharmaceutical companies. There are several things, analytes, that people have developed assays for to measure that have different potential applications depending on what type of studies you're interested in. Integrated HIV or proviral DNA is both the largest magnitude and the easiest assay to measure with. 2LTR circles, which are an abortive integration process, have some potential utility for some certain purposes. Cell-associated HIV RNAs are transcripts that we want to induce, so that's going to be important looking at interventions. Some people are interested in looking at plasma RNA as a measure of an impact on the reservoir. The quote-unquote gold standard is the inducible infectivity by limiting dilution co-culture, because that measures actual replication-competent virus, because what we know is most of the provirus is not replication-competent. And the ultimate standard is no detectable virus after you withdraw ART and watch the patient for many years. And we have one such success out of the 79 million people who've been infected so far. And that's at the Berlin patient. So Matt Strain in our group applied the droplet digital PCR approach to measuring DNA and RNA, which provides more precision and sensitivity at the lower end for looking at rare events than does RT-PCR. And looking at between 1 and 200 patients who are fully suppressed, in black you see the type of results you get with RT-PCR, and in blue the results you get with droplet digital PCR looking at 2 replicates. And the duplicates are obviously closer and more reliable with the blue than in the black, showing the improved sensitivity and precision of droplet digital PCR. But no matter what assay we use, the reality is that, let's see, is this the pointer? If we take a milliliter of blood or 1,000,000 cells, these are the magnitude of what we see proviral copies in a patient who initiates treatment with chronic infection. You have between 10 and 1,000 copies. In terms of infectious units, maybe 1 to 10 in 1,000,000 cells. And plasma viremia can be anywhere from completely undetectable to maybe up to 10 in a fully suppressed patient. If you take then 10 mils of plasma or 10,000,000 cells, this is a dynamic range of an impact that you can measure if you apply a candidate intervention and then are trying to measure something. But having a dynamic range of at best 10 or 100 or 1,000, once you get below that range, this is what you have to document or actually achieve in order to cure a patient. First, we don't know when for reducing proviral copies, which are the easiest to measure, which proportion are replication competent. And finally, once you get below the level of detection, you haven't cured someone. You just can't detect them anymore. And despite a lot of publicity and publications, there's a fundamental principle that the absence of detection is not detection of absence. So the standard gold, or the gold standard for assays are to take PBMCs, purify the resting CD4s from them, do a terminal dilution assay, induce those cells with some sort of activating agent, and then look at viral outgrowth in the individual wells and do a binary present or absent assay, and then use Poisson statistics and other approaches to measure the presence of inducible infectious virus. But what Bob Silicano's group showed, Yo-Chi Ho did a beautiful study in which she took the cells that did not grow out virus and then did a full-length sequence of their provirus and showed that the vast majority of them either had deletion or various hypermutations which are consistent with the lack of replication competence of those. You could completely predict they wouldn't be replication competent, but suggested that as much as 16-17% of them were, at least by sequence, potentially replication competent, and they sort of confirmed several of those, so that represents at least an order of magnitude more replication competence than we get with the gold standard assay. And these are the gold standard assay is in yellow. The purple is the magnitude of the proviral reservoir, and this pink is what they project as at least theoretically replication competent. And so what we have to do is figure out better, cheaper assays. And Marta Massanea in our group has taken a lot of nice approaches to try to do this. Once again, enriching purified resting CD4 cells, and then modifying the viral outgrowth assay by doing the same thing, serial dilution, induction, and then putting in cells that are as sensitive in our hands as CD4 cells in the standard viral outgrowth assay, and measuring the propagation of virus. So this is now a 7 or 8 day assay that is, in our hands, more sensitive than the viral outgrowth assay for detecting virus. And it's a 1 week assay. And part of the sensitivity is achieved by a better way of measuring viral generation. And I won't go through the details in the interest of time. And she's also looked at another variant in which does the same serial dilution, but puts raltegravir in the system and induces with anti-CD3, anti-CD28. And so the generation of both transcripts and virus is only in the cells that are induced, and there is no ongoing propagation, so that this assay only takes 3 days. And what you can see is this assay, which may be a log better than the standard viral outgrowth assay, detects this. The measurement of induction of transcription is wow, that was impressive. Is even more sensitive, and we're starting to get much closer to the proportion that would be predicted by the Silicano group of potentially inducible proviral copies. And so applying these now to various clinical specimens is what's being done in our group. So I will stop here. Looks like I've broken the whole process here. So that's the end of the symposium. Oh, there we go. So I want to acknowledge various colleagues and collaborators who it's been a real pleasure to work with. The folks who are emboldened there are the ones who've specifically developed the assays that I just talked about. But just like with the HNRC, it's really great to work with a large number of talented colleagues and to have a lot of terrific collaborators outside the institution as well. So I'll stop here and thank you for your attention.